All right, here we are for another biology video. This one, as you can see, is going to wrap up the first week of the second semester, which has been about biotechnology. And in particular, I want to talk about genetic engineering, because this is kind of where we left off from the first semester. At the end of the first semester, you did a project about designer babies, about being able to manipulate DNA to control the traits of future hypothetical offspring. Right? And so, how possible is that? How would that actually work? Could we actually do it? This picture that I got here on the screen, it kind of makes it oversimplifies things. It makes it seem like you got the scientists in there just like changing the DNA or whatever they want to do. But it's a lot more complicated than that, of course. But we are getting closer and closer to the ideas of movies like Gattaca or Twins, even, where controlling traits is possible. So let's start with a trait like diabetes that you would maybe want to engineer your baby to not have. But we don't have to engineer babies to help with diabetes, right? Diabetes is caused by high blood sugar, which is in type 1 diabetes, which will be the kind that we're going to talk about today, is due to a lack of insulin. For a variety of reasons, your body just doesn't make insulin. Insulin is supposed to make your blood sugar go down. So when your body releases insulin, it tells your body to take the sugar from your blood and convert it into this stuff called glycogen, which is stored in your muscles and liver and fat cells. But it is a protein, right? And so we know that proteins are created by our cells by ribosomes, and the instructions for proteins are in our DNA. So especially for type 1 diabetes, the problem here is actually a genetic problem, that there's something wrong with the genes. And so that means that we could use genetic engineering to try to solve that problem. Now, before we had genetic engineering, we would just give people injections of insulin. People still do take injections of insulin, but not too long ago, your insulin might have come from a different animal, a pig or a cow, or even, believe it or not, a shark. Sharks have insulin, like all animals have blood sugar that they need to control. And so sharks or pigs or cows, they have a lot of the same proteins as we do, but they're not exactly the same, right? So down here's the human sequence, just some of the amino acids, right? Hopefully you'll remember the 3-letter abbreviations. You don't have to remember what they mean, but that these are in fact amino acids, right? Porcine here means pig, and you'll notice that the pig insulin is exactly the same except for right here, which is at the 30th amino acid on the beta chain, which you don't need to know that. Cow's not as good. The cow has 3 differences from the human, right? So it doesn't work as well, but it still does work. I don't have any information on the sharks. I would imagine it would have even more differences than the cow did. But nowadays, you're not going to use pig or cow insulin. You're going to use insulin that was created by transgenic organisms that have been genetically engineered to produce human insulin, right? So they would actually make the real human form of insulin with all the same amino acids. Although, now that I say that, I'm about to mention that this particular brand of genetically engineered insulin is actually slightly better than regular human insulin. They have found a mutation right here that actually makes the insulin more effective. So people that get this insulin that they inject into themselves to replace the insulin that their body doesn't make, they actually get slightly better insulin than those of us that have a normal working insulin gene and everything like that, which is kind of interesting. So we're going to use, most of the time, bacteria or yeast, but I'm going to just go with bacteria to do this kind of thing, right? Because it's a protein that's produced in the body, we can find the chromosome, number 11, where insulin gene is found. We can cut it out and put it into a bacterial cell. And then the bacterial cell will make human insulin that we can harvest and give to people, right? So how does that work? How do you genetically engineer something on that level? Well, first you got to cut out the gene. To do this, we use these enzymes called restriction enzymes. They are proteins that cut DNA at specific places, right? Kind of like molecular scissors. They sit on top of the DNA and they cut the backbone in a couple places, right? So then you have this, the DNA has been cut up. We know of thousands of these enzymes. We get them from bacterias and fungus and things like that. All kinds of different microorganisms have these enzymes in them naturally, and we have found a lot of them and figured out where they were, how they cut. This is just a brief diagram that shows that these are all the names of these restriction enzymes, and these are the places that they cut, the sequences. If they find this particular sequence, they will cut in that particular spot, right? So what does that mean? Here are two of the most common restriction enzymes. They are called EcoR1 and HindD3, and they have their particular spot where they are going to cut, right? That whenever they find this sequence or that sequence, they're going to cut. Now, the dash means it's going to cut between the G and the A, or in here, in between the A and the A, right? So you would cut it between the G and the A on both sides, or between the A and the A on both sides, and those pieces are now free. They're disconnected. They've been cut, right? Like that's the whole point of the deal. It leaves these little overhangs that are sometimes called sticky ends, which means that these letters would like to bond with the complementary letters, and so they want to just go back together how they were, but we can jump in there in the middle of that process and make them connect to what we want them to connect to. So for example, let's say we have this long string of human DNA, and we know that the insulin gene is on chromosome number 11 in this one spot. Well, we find a restriction site on either side of the gene, right? So here's a restriction site on the left, here's one on the right, and if we can just cut it with the restriction enzyme, we will end up with a fragment. We'll end up with this piece that has the insulin gene on it, right? So now we can take this little piece and transfer it to wherever we want to transfer it to. We want to get it into a human, or into a bacteria cell, rather. How do you get it into a bacterial cell now that you've cut it out? Well, bacteria have these little things called plasmids, the blue ones on the side. They're separate from the main piece of DNA, and they are specifically used to transfer DNA between bacteria. They are kind of completely, like it says, independent of the rest of the cell. And so if you have two bacteria cells and one of them has a plasmid and it wants to give it to the other cell, they just link up, and then the plasmid gets transferred into the other bacteria. And so now the other bacteria has a copy of that set of DNA, right? You can use viruses and yeast. We'll talk about that in a minute. But plasmids are the most common way that we would transfer DNA from one place to another. So the top line up here, this is the human DNA, and the bottom line here that I'm showing you is just a part of the circle for the plasmid. We want to cut each with the same restriction enzyme, right? The same sequence, hopefully, is found in both of them. And then we want to hopefully get the insulin gene to connect to those two fragments, right? Right there, matching sticky ends. They would hopefully join and make a solid piece. Now, I do have to point out at this point that this is not nearly as nice and neat as it makes it seem here, that you have all these pieces in your test tube, and they could combine in any number of ways, right? Here is a spot where you could connect, and it could join with that piece, right? This piece could join all the way over with that piece, right? There's all these different combinations that you can make because you have the same sticky ends on all the sides. It is a numbers game. You know that it won't work a lot of the times, but hopefully it will work enough times that you'll get these sum of these plasmids that would look like that bottom thing, right? And then you would isolate those through a number of techniques, of course, because you don't want all the other random pieces that got made. You want only these plasmids. But now that you have the plasmid, it has a piece of human DNA inside of it. We can now use the plasmid to transfer that into a bacteria. So you take your bacteria and there's a few different strategies that you can use. You can use chemicals to force the bacteria to take in the plasmid. This was actually the job that I had when I was first out of college, and I used electricity. I would shock the cells a little bit to get them to take in the plasmid. If you were going to take AP biology a couple years from now, you would use heat to make them take in the plasmid. And yes, that means that in AP biology, you get to do this for yourself, right? So however you're going to do it, you're going to get the plasmid into the bacteria. The bacteria now is going to use that DNA, right? The bacteria doesn't know the difference between human DNA and bacterial DNA. They're all just A's, C's, G's, and T's. And so once it's inside the bacteria is going to start to make, in this case, human insulin or whatever gene that you wanted them to make the protein from, right? So first, you cut out the gene that you want. Then you cut the plasmid with the same restriction enzyme. You mix it all together, and you make this thing called a recombinant plasmid. You take that and you put it into the bacteria. You find the bacteria that took in the DNA and you grow them until you have this big container, and basically the bacteria grow in a liquid, and the liquid then will contain a bunch of insulin because the bacteria will be making the insulin, and the insulin will just go off into the liquid. And so you have this liquid full of insulin, and then you process it and you give it to people, right? And so genetic engineering has allowed us to make all kinds of different proteins by using bacteria or yeast and using plasmids. But that only works for things like diabetes, where it's just one certain protein that you're lacking, and you could inject that into the person, right? But that doesn't cure their insulin. That's just a treatment for the diabetes. Sorry, I meant diabetes. It's not a cure for diabetes. It just treats the diabetes. What if you wanted to actually design a baby so that they wouldn't have diabetes? Well, this is much more complicated. You can't just put a plasmid into a baby and it starts working. You have to actually change the cells, right? At this point, you wouldn't just be messing around with one protein. You would have to change every cell in that baby's body, which means you have to do it really early, like in the egg, or even in the egg of the sperm, or in the embryo. But that would mean you need a better way to edit the DNA. You need what's called precision gene editing. You need a way to cut the DNA anywhere that you want. That would let you mutate or delete any gene if you wanted to do that. Like maybe you had a harmful gene and you wanted to just knock that out and make it not work anymore. You could do that. But more likely, you would want to cut the DNA and insert that gene, right? Instead of putting the insulin gene into the plasmid, you want to put it directly into the person's DNA. To do this, you would use something that has been developed over the last 20 years or so, called CRISPR, which you might have heard of before. It is getting more and more and more and more common. And in fact, at this point, all scientists would rather use CRISPR for their job rather than using a plasmid, because CRISPR's much more direct. CRISPR has two parts, a guide RNA. So let's say that down here you have a gene that, in this case, a gene that you wanted to break. You would make a guide RNA that would match perfectly with the gene that you wanted to target. Then you would add an enzyme that will cut the DNA. This is the Cas9 part. The Cas9 is an enzyme that cuts DNA. And so this little complex will then go and attach exactly to the spot where the guide RNA says to go, and it'll cut right there, right? It'll just break the DNA. Here's a different picture that I like on a number of levels. One, it's simple and it shows exactly how it is. You have your RNA guide that's inside of the Cas9 protein. I do personally like that they made the Cas9 look like a little teddy bear that is giving the hug of death to the DNA right there and cutting it, right? But if you cut your DNA, it's broken. Now that gene won't work. And so again, this isn't what you would do for insulin. You wouldn't want to break the insulin gene. In fact, the person that you'd be doing this on, their insulin gene probably already doesn't work. But you would want to do this in a number of other cases, again, where you would want to get rid of a bad gene somehow. But there's this interesting thing that happens, is that when you cut the DNA, it makes this little space, and the cell doesn't like that space being there, and so it wants to repair it. It will always try to fix the cut that you made. And that's okay usually, because again, when we're trying to break the gene, when it repairs it right here, it's just going to put any number of DNA letters in there, A's, T's, G's, and C's. It's going to be random. And so it's not going to make any sense. And so this is basically introducing a huge mutation. But scientists found that if you include another piece of DNA, when you make that cut, the cell would rather use the already made DNA to go into that hole rather than just put a bunch of random letters. And so, most of the time, and it's not all the time, and that's a problem, you can cut and, in effect, paste a new gene into the spot that you want it to be. And this means you could introduce any gene that you wanted into any animal you wanted or any organism you wanted at any place for any gene. Now, right off the bat, this is totally illegal. You might have heard that somebody from China tried to do this on a couple babies and got in a lot of trouble, because for a few different reasons. The very first reason is that this isn't perfect. And when you make these cuts, most of the time, the donor DNA goes into the spot, but sometimes it just makes weird mutations. Sometimes it accidentally cuts in the totally wrong place. There's a bunch of different things that could go wrong. Now, most of the time, it doesn't do it wrong, but it does happen enough that it wouldn't be safe to really use on a person. We don't really know about those two babies from China and what happened to them, or how many times it didn't work before that, right? And so this is definitely not safe at the moment. The second reason would be that if you start messing with people's DNA, you then mess with their DNA, right? Now, if you change the DNA of a baby, when that baby grows up to have their own babies, that same change will be in their own babies, right? Like, this is going to permanently change somebody's DNA, as opposed to, again, the insulin, where you just get an insulin shot and it's temporary. So there's a lot of things to think about when you start to talk about advanced gene editing. Of course, there are a lot of movies now being made about the dangers of gene editing. And so that gives me back to this picture, right, where the common person kind of has this idea now in their head that, oh man, there's scientists in there just messing around with DNA and cutting things up and putting things in other places and taking the DNA from this organism and putting in this other organism, and it all sounds kind of science fiction-y. I like this picture just because of the ridiculousness of it. A banana fish obviously isn't real. This isn't what genetic engineering is. And actually, I don't know if you know this or not, but you already share 40% of your DNA with a banana, right? So if you're going to go from that way of defining things, you are 40% banana. That's not really the right way to look at it, though, because again, this is DNA, and they're just instructions for making proteins. There's no such thing as a banana protein. A banana is a big old group of a whole bunch of different proteins that all work together to make a banana. And like we've shown with the insulin, you can take those genes and move them from one animal or organism to the next, and it doesn't really change the actual organism. Now, most of the time, historically now, these organisms have been used to produce very simple things, again, like insulin or growth hormones, blood clotting factors, all kinds of things. If there's a protein that a person's body isn't going to make for some reason, we can make it for them using transgenic organisms, genetically modified organisms, right? These medicines are out there right now helping millions and millions of people. We can use them to make vaccines. We can take the information from a harmful virus like the hepatitis B virus, one that you wouldn't want to make a vaccine for because it's too dangerous. But you could take some of the DNA for the spike proteins, for example, from the hepatitis B, and put them into a harmless virus like the cowpox virus. Now you have a harmless virus that looks like the harmful virus, and you can inject this into the human to get their immune system to react to it, right? And you can be protected from hepatitis B by getting this genetically modified cowpox virus. A lot of what you watched last week and what you read about was about gene therapy, right? So hopefully it makes sense to you now that we would take somebody's actual cells out of their body, modify them, and then put them back into their body. And hopefully this would allow the cells to act normally and alleviate or even cure whatever symptoms are happening from their genetic disease, right? Like the eye disease from the video. Um, this is right now the most advanced technique that is actually being used, right? Out of all these different things that you might have read about, gene therapy is the most advanced one, the one that has been developed the furthest and is the most practical. Um, Another thing that you read about would be cloning. And of course, people, when you think of clones, at least people from my generation, and maybe just me and my friends, because we love Star Wars, we clones, right? Like it's a clone army. And people are worried, like, oh, clones, you could use the clones to make a clone army. Or then there's the students who have this idea. I don't think they really have this idea, but there's that idea out there that, like, oh, I can make a clone, and my clone will do my chores and my homework for me while I go and play. And that you could make a clone, but it probably wouldn't work out the way you think it is, because you don't just get a bunch of clones. What you get are a bunch of babies, because cloning is just having a baby normally, but it's been genetically engineered at one critical step. Take the egg cell from the donor and remove its DNA. It would basically a blank egg cell. Take just the nucleus from some other cell and put it into the egg cell. Now you have this egg cell that kind of isn't an egg anymore. Now it's kind of what's called a zygote. It's a cell that has a complete set of DNA. And if you give it the right chemicals, it will start to grow into an embryo. Then you need to put the embryo into a female. We do not know how to grow an organism. You might have seen something or heard about they did make an artificial uterus, and they grew a baby sheep, a lamb, for like 2 weeks inside of this bag before they freaked out and put it back into mom, right? But it was by no means did they actually grow something in the laboratory. At this moment, and probably still for a very long time, the only way to get a new organism is from a female organism, from a uterus that will grow the embryo properly, right? So in the end, you get a clone of the original, but the clone's a baby, right? The clone is not the same sheep. It's more like an identical twin than anything, right? The sheep would not have the same experiences as the original sheep. It wouldn't be the same sheep. Right now, it is possible. You can pay a lot of money. We're talking lots of money, and clone a pet like your dog or your cat, right? And I think that some people think this would be a good idea because their favorite pet that they had for 15 years dies, and they're like, oh, I want Lassie back. And they go and they take Lassie's hair and blood and they give it to the lab, and they use that to make a clone of Lassie. But that's not going to be Lassie, right? Like, that's not your dog anymore. That's another dog that looks exactly like your last dog. But that dog doesn't know you. That dog doesn't remember the time you went to play fetch in the park. That dog is a brand new dog, right? Like, a clone is not the same thing as what, again, in like science fiction movies, you're led to believe they are. But people aren't cloning that much out there. Just about every animal has been cloned, but this isn't actually something that you run into on your daily life. But the most common way that you interact with genetically modified organisms in your daily life is through food, although these aren't food. Those are called glowfish. Those are fish that have been genetically modified to glow, right? But we can genetically modify just about anything, right? This is kind of the idea. Perfume that was made by yeast, right? Instead of actually making it from flowers like you would traditionally make it from, you could take the genes that make these chemicals, right? These chemicals are what make the flowers smell a certain way. And those chemicals are made by enzymes, and the enzymes are found from genes. And so you could take the genes and put them into yeast, and they will make yeast that smell like flowers, right? But this would be weird to some people, right? Like genetically modified perfume, let alone genetically modified food. And so the underlying idea is that you could make any trait. If you knew the gene, you could control any trait, right? Like you could make something taste sweeter, make it have a different color, make it bigger, make it not get bigger, but grow to its normal size faster, right? And then people take this and extrapolate it to like, oh, we could make somebody be musical, theoretically, right? If you knew the gene. And this is what some of you guys were looking up. Like, if you know the gene for it, you could put the gene into whatever you want it and make it the way you want it. But it is way more complicated than that. And in the end, genetically modified foods are rather tame. Um, they make them resistant to herbicides, which would be the chemicals you spray to get rid of the weeds. Right. This is something like Roundup is a chemical that you spray on weeds. It's a weed killer. But then you could genetically modify your plants, like your corn plants, so that they don't die when you spray Roundup on them. And so then you can just spray Roundup all over your plants, and you don't have to worry about them dying, but the weeds will die. Sometimes the plants will just straight up taste bad to insects. Some of them even make things that kill the insects, right? So you can make the plant produce a protein that kills insects, not humans or other animals. You could make them produce more vitamins. There is something called golden rice, which rice does not normally contain the vitamin called vitamin A. And so they have genetically engineered rice so that it will produce vitamin A. And so now when you eat the rice, you will actually get more vitamins than if you didn't eat that kind of rice. Um, there's tomatoes that will stay firmer after ripening. There are a whole host of things, of mutations that they've made or changes that they've made. The most common ones are for corn and soybeans, mostly to make them resistant to pests. Potatoes that will grow faster and bigger, right? These are a thing. But and these are pretty much the only kinds of foods that are currently on the market that have been genetically modified in any kind, right? So when you see labels on, say, on the bread that says that this bread has not been genetically modified, well, of course it hasn't, because there's no genetically modified wheat out there, right? Unless they used genetically modified corn to make the sugar that they used to make the bread. But still, again, that's not the same thing as genetically modified bread. Because genetically modifying things just means taking a gene from one other organism and putting that into a new organism, right? For example, you could take a gene from this thing called an ocean pout, which is some kind of fish, and add it to a salmon so that the salmon will grow faster and bigger, right? You can see on this particular diagram the comparison of the normal salmon compared to the genetically modified salmon, and the genetically modified salmon is way bigger, right? So this is good for the producers of the salmon and for the consumers, because theoretically this means that it would be cheaper because it would be more supply out there. But is this really going to happen? It hasn't happened yet. The genetically modified salmon have been around for years, but you can't really buy them yet because people are afraid. People maybe are not ready for genetically modified animals or organisms. Again, because maybe they don't understand what it means, because they're thinking back to the banana fish and they're thinking, oh my gosh, a genetically modified salmon, like it's a frankenfood. It's a monster. It's unnatural. Now, I don't know about all those words like unnatural, but DNA's plenty natural, and it works in all sorts of situations. Ugh, that was awesome. And in the end, again, all you've done is taken a gene and moved it around. It doesn't change the actual animal. It just means that the cells of that animal will produce one extra protein than they normally would. But are there any actual concerns about genetically modified organisms? And yes, there definitely are. This isn't something that we're just going out there and modifying everything around you and just like, who cares? The first concern would be the effect on health, in particular, allergic reactions, right? No one's ever eaten this particular strawberry that might now have an extra protein in that would make it be resistant to freezing, this is a thing that strawberries, if the environment gets too cold while they're growing, they will freeze and then they'll die. But if you could make them resistant to the cold, then they will survive the colder temperatures, and you could grow them in more areas of the world, right? But that means that inside of that strawberry, there's an extra protein floating around that no one usually eats when they eat a strawberry. So maybe that would be weird. Maybe that would cause an allergic reaction. But at the same time, The place where they got that gene was from a fish that people eat, which means that people eat that protein all the time. And so there shouldn't be any more allergic reactions than there already was. But of course, even if that wasn't the case and that this was actually a protein that really nobody had ever eaten before, they would just test that, right? Like they would have the procedures to regulate those things to make sure it was safe before they started selling it to people. So negative reactions when consumed are not really realistic because of the nature of what it means to genetically modify something and because of the nature of how we have safety procedures to make sure that things are safe before you go out and start eating them. The real danger would be on ecosystems. What if they escaped? What if those giant salmon got into the river? Are they going to eat all the food and all the little salmons, the natural, the native salmons? Are they all going to die? Right? Like, we don't know that. What if the corn that has been genetically modified to grow faster, if the seeds get out there and they start growing out in nature and they are able to reproduce with some other corn and make like some crazy version of corn we've never seen before, right? These are real things. You definitely need to be aware of how our actions are going to affect the ecosystem as a whole and not just our bodies in particular. But as long as we have safety measures, again, as long as there are assurances and methods used to keep the genetically modified foods away from the natural foods, then this also isn't a huge concern. And again, going back to the fact that the concerns mostly rely from what you're going to eat, there are no concerns about what it is when you're going to eat, right? To the point where a lot of these things are just overblown because people watch too many sci-fi movies, just like the cloning, or just like how using viruses to cure cancer ends up killing the whole world in like The Walking Dead and stuff like that, right? Like that's not really how science works. So in the end, genetically modified organisms have and will continue to have a lot of benefits for us and for the environment. Of course, we are still developing them and there's going to be some hiccups, but these are not things to be afraid of. And as we go talk more and more about genetics through the second semester and about evolution, maybe it'll become more clear about the role that genetically modified organisms are going to play in our world.